This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1075, recorded on December 29th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. A brief excursion into the weather report says that if I closed my eyes and I didn't know what date this was, I would swear that I was getting out my fishing equipment and waiting for the trout season to open. It's mild. It's beautiful. It shouldn't be this way in December. That's very off-putting. Very off-putting.
Get used to it.
Yep, you're right on that.
Near New York is 12 degrees Celsius and mostly sunny.
Right.
Yeah, I wore a scarf today, totally unnecessary. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 41 Fahrenheit, which is 5 Celsius, and it's drizzling, and it's been gray and cloudy for more than a week. We had a foggy Christmas, and we might have sun tomorrow, they said.
Aren't you due for some snow out there?
They said we might have snow on Sunday, but it won't stick, won't accumulate.
It won't stick. Yep.
They don't say stick, they say accumulate.
Accumulate. I'm thinking of,
yes.
What we're going to say today is not going to stick, probably. All right. Also joining us from Madison, New Jersey, Brianne Barker.
Hi. Great to be back. It's 51 Fahrenheit, 10 Celsius here. And it is currently not raining. It has been raining for quite a few days straight, which would have been quite the snowstorm in other years. But does not feel like December.
Nope. And from Montreal, Canada, Angela Mingarelli.
Hello. Welcome to the last TWiV of the year. Well, here it is actually above zero, which for December 29th is kind of terrifying for Montreal. There's no snow outside. Those that can see out my window. It's 1 degree, and it's kind of like raining / almost snow. It's like a wet snow. But yeah, we don't actually have any snow. I was in Ottawa for Christmas, outside of Ottawa, which is about a 2-hour drive, and we did have snow there, but it was only a little bit, and it's actually pretty sad. Like white Christmas, like that's the best part of Christmas is the snow, at least for me.
Now we're gonna have fake snow. Like we have fake news and fake everything in the world.
So one of the big worries that I've been reading about off and on for the last year and a half maybe is the melting of the permafrost above the tundra. And what it's going to release, it's not just releasing methane gas. It might be releasing microbes that have remained dormant for some 40,000 years. And
it might.
I wonder what might happen.
We'll see, Dixon. Hard to predict, isn't it? But you said the other night that Google wants to build a data center up there in the permafrost.
Yeah,
right?
They claim they do.
That's smart. Why don't we put heat where it's not supposed to be?
Exactly right. That's right. Or use the goddamn heat.
Dixon,
I mean, come on.
Dixon was on a live stream Wednesday night. You can
time.
find it on YouTube. It's recorded. He talked all about his book, The New City. I think we got a few book sales for you, Dixon.
I did. No, I was very pleased with that. I could just
hear
that old cash register going, how does they sound again? I think I've forgotten.
There's a guy who, he was on a podcast, Steve Gibson, Security Now, and he has a program that he sells, and he had an app on his phone that would make a cash register sound every time someone bought the app.
I love it.
So he'd be recording and it would be going cha-ching, cha-ching.
Oh, that's how it sounds. Right.
If I had that here, we'd never hear anything.
No, it'll be dead silence. Exactly right.
Anyway, I don't want to complain about that. if you enjoy what we do on these programs, and as Angela said, this is our last TWiV of 2023, although this one will drop on Sunday, so last day of the year. Please consider supporting us. Many of you have started supporting us. It's great. It's a way of you to feel good about something that you like. Right? Go to microbe.tv/contribute. And Kathy has an announcement.
Yes, as Vincent said, this will drop on December 31st, which is 123123. And before very long, we will be in the something 24, which is when the ASV 2024 meeting will be in June. And February 1st of 2024 is when your abstracts are due. And if you are going to apply for a travel award, you should think about doing that because the deadline is February 3rd. And you can get all of this information at asv.org. The thing you should do now, besides finishing getting your data for your abstract, is join ASV or renew your membership. Renewed memberships are due basically as of January 1st each year. So if you're going to apply for a travel award, you need to be a member if you're a student or a postdoc. So now would be a really good time to renew that membership.
Oh, I have to do mine yet. Oh,
we talked about this last time, Vincent.
I have to write it on a sticky. Let me do that right now.
You're reminding me. I'll be doing mine after this as well.
Are you writing it on a sticky?
ASV. By the way, look we have now Microbe TV stickies.
See? Whoa! I don't have any of those. Yeah.
I can send you some. We bought 250.
I have Parasites Without Borders stickies.
Oh, nice.
Competing stickies. These are very cool. And I will send you guys all some next week. They're cool. We bought a bunch, and I got renewasv.org on this one. So the key is I have to
look at it.
I don't know where to put it. Right in front of the keyboard here.
Yeah, I put them on the bottom of my screen for my monitor. That way I don't forget them.
Yeah, that's a classic place to put it and not look at it. Yeah,
right.
And there's a good place to put all your passwords so everybody can see them.
That's right. Exactly right.
All right. We just to remind you, there's Amy Rosenfeld at the FDA still looking for another research assistant to work on enteroviruses like enterovirus D68, poliovirus, human rhinoviruses, and to do all sorts of genetic and biochemical and cell-based studies, and for understanding antigenic antibody cross-reactivity among the viruses, what consequence that has to make animal models, all BSL-2 work. And if you go to our show notes at the address for this episode, microbe.tv/TWiV, you will find a PDF. You'll have a little more description of the job and minimum qualifications, and Amy's email, so you can email her. And also we'll remind you that Dixon has a new book, The New City, as we already mentioned, and you can go to despommier.com for various ways to buy it. It is really important to get the word out. Dixon has said that he will give all profits to charity. Right, Dixon?
Exactly right.
He just wants the word to get out there.
Remember, charity starts at home.
That's right. Despommier.com. Let's see if it works. I mean, I had it.
I just checked. It does.
Oh, good.
It works. We should change. Please consider making a contribution to. Why don't you take us out for a cup of coffee? I love that little tag at the end of that one email that we got. And she said, that makes it sound so friendly. I would love to take you out for a cup of coffee.
But I want more than a cup of coffee.
And a donut and maybe lunch.
No, a cup of coffee, many people buy coffee every day, right?
Friendly gesture.
And if you go to a fancy coffee shop, I won't name any, it can cost you $3, $4, $5, $6, $7, $8, right? So you get the fancy stuff. Nobody buys black coffee anymore like I do.
Nobody buys coffee. I don't even know what that stuff is that they're serving.
Anyway, it's very expensive. And if you just gave one of those a month, right? $3, $4,
that is the way to put it.
Buy us a cup of coffee a month. Yeah.
That's right.
Microbe.tv/contribute.
Might even keep us awake.
Now, folks, for this last TWiV of 2023, we have 2 tough papers, okay? But we're going to try and simplify them a lot. And because Angela's here, I decided that the first one should be about chickens. Do you like chickens, Angela? Not to eat, but just to, you know.
I mean, I consume chickens, yes, poultry. I consumed a lot of turkey on Christmas.
But do you like chickens as animals, as nice farm animals?
I mean, they're
No, you
fine.
don't.
I
They're
prefer
fine.
cows to chickens, just because chickens are they're a lot of work. Well, they're not, and they're really smelly. They smell terrible. Like a
Really?
chicken coop, yeah. Being intensive in like chicken farming, it's pretty, it's an aggressive smell. But no, chickens are fine. They're not my favorite animal. I would say I prefer cows if we're going to talk
Cows.
yeah.
Because you could put your arm in a cow, right?
No, not only they're just so sweet. Cows are actually like the sweetest animals. We spoke
Really?
about this.
Yeah.
Yeah, they're very like docile. They won't kick you like horses, or very rarely will they kick you. They don't really bite.
But you told us
They're
on
nice.
TWiV a while ago that you have to be careful where you stand behind a cow when you're putting a harm in them, right?
Yeah. Oh, for sure. But horses are like 50 times more scary. Cows are they're super chill compared to a horse.
That's funny.
You should
Cows
watch
are-
that series, All Creatures, Great and Small. There's a lot of scenes in which he has to put his hand down the backside of a horse or a cow, and occasionally he comes home bruised, not just his ego, but also the rest of him.
Yeah.
It's quite a challenge, and it's not trivial. And people sort of trivialize, oh, you're a vet, you deal with animals. That's doctors. Your patients can't even talk to you. Well, maybe the gray parrots can talk to you, but very few other animals have that gift. So you have to be a better detective.
A long time ago, one of my picks was this YouTube video series by Martin. Forgot his last name, but he bought some old stone houses up in northern Italy, and he's renovating them, and he shows videos. It's pretty interesting. So recently, he bought some chickens, and he has a coop outside, so it's not smelly, right? Because it's outdoors. And during the day, he lets them walk around, and they just make these funny sounds. They're like, walking around.
That means
It's a good impression.
There's a hawk flying above us. There's a hawk flying above us.
It's an interesting thing. You should say that. But he puts them away at night because that night the foxes come out, and in the morning he can see the fox tracks. They're trying to get in the cage, but there's no way that they can get in.
Right.
And unfortunately, he had originally 5, and I think 3 of them got killed in various ways. One, some visitors came with a dog, and the dog ran after the chicken and killed it.
Oh, no.
Really?
And it bit the tail off the other one. There's a white chicken. It bit its tail off. And so now that chicken doesn't want to come out of the coop anymore. But anyway, they I find it interesting, the sounds they make. And they like to hang around him, right? When he's doing stuff, they like to hang. It's almost like they like people, right? And they lay eggs. Every week he gets eggs from them and makes omelets.
Every day if you're lucky. If it's a healthy chicken,
every day
it
if
should
you're
be
lucky.
laying every day. Yeah.
Okay.
We've done a lot of traveling.
So-
I'm sorry. Go ahead.
This first paper is about chickens. So it is a paper in Science, and it's called Ancient Chicken Remains Reveal the Origin of Virulence in Marek's Disease Virus. Here in New York City, you always see chicken bones on the street, in the subway. People eat them, and then they just throw them away, right?
I think these are slightly more ancient than that.
But these are more ancient.
Paleo bones.
So we have 2 co-first authors, Stephen Fiddaman and Evangelos Dimopoulos. And then we have 3, I think, senior authors who contributed equally. There are a lot of authors in between. Gregor Larson, Adrian Smith, and Laurent France. These are from all over the place. University of Oxford, Université Toulouse, ETH Zurich.
A lot of museums.
And archaeology departments. I'm not going to mention them all.
No, but there's some from the Pirbright, and my favorite one, because it's where they got their controlled chicken, was the Silky Club of Great Britain from Charing, UK.
That's good.
Silkies are a kind of chicken.
Vincent,
and there
is this
Silky clubs all over the world?
Huh.
Right. Is this the tumor that was first identified by?
No.
No.
No. That was a sarcoma. Had
That's
nothing to
right.
do
That's
had nothing to do with Marek's
so cool.
disease
That's right.
virus, which is a herpes virus. It had to do
Okay.
with an RNA tumor virus, Rouse sarcoma virus, right?
Indeed. That's right. That's right.
Did you meet Rouse, by the way?
I did.
Okay.
And I actually saw the section of the tumor also.
Cool. All right. So Marek's disease virus
Mhm.
is a herpes virus that causes tumors in chickens, and it was first described in 1907, called Marek's disease, and it was at the time a mild disease with low mortality, but over the years the mortality increased until now it's over 90% mortality in unvaccinated chickens. And so the poultry industry has started vaccinating a long time ago to prevent this virus from destroying their chickens.
It's cool. The
And they say,
vaccination is actually in ovo. So on day, I think it's like 18, it's actually they vaccinate like the embryos within the egg, and it's all like in industrial farms. This is done like mechanically. It's not even done like by a person. It's literally just like injected into the egg, which I think is interesting.
That is really interesting.
What is the nature of the vaccine?
Oh, there's multiple. There's many Marek's vaccines. There are what is it? Inactivated. I think the Rispens vaccine. It's called CV 1988 Rispens. It's an attenuated vaccine of Gallad alpha herpes virus 2 because there's multiple serotypes of Marek's. They also vaccinate with one of the turkey herpes viruses as well, which is what is it called? Meleagrid something? Meleagrid alpha herpes virus 1. And then they have multiple recombinant vaccines as well with Newcastle infectious bursal disease virus and infectious laryngotracheitis virus.
Wow. So was this
two
one of the
birds, one
first
stone.
was this one of the first effective anti-cancer vaccines?
That's a good
I don't
was
think
the
you can
vaccine?
call it. I don't think you can call it an anti-cancer vaccine because it's against the virus, and yes, the virus can develop into. It can be oncogenic, but I don't know if it's right to say anti-cancer. I don't know
Well,
if
Well, if
defining that.
it prevents infection or
Yeah.
disease. Let's say it prevents, disease.
so the chicken doesn't die of a tumor; it dies of the disease that the virus causes.
No,
They
it
also
doesn't
have-
die, right?
No, they don't
So-
die. Or if they do, there's neurological deficits a lot of the time. So I remember in vet school, like the main thing you learn when you learn about Marek's is that they have like this extremely enlarged sciatic nerve, like the sciatic nerve, the vagal nerve. But the sciatic's the easiest to see in necropsy because you just like open up the thighs and then you can see it. It's huge. It's like 5 times the size it should be. And they have like proprioceptive deficits. They have ataxia. Their legs. It's very typical to see a bird with like paralysis, leg paralysis. With Marek. So that's like the pathognomonic clinical sign is like birds with paralysis and proprioceptive deficits.
They
Hopefully
kind of
vaccinated, but
yeah, hopefully vaccinated. They kind of hint at this in a couple places in the paper, but how is the virus transmitted from chicken to chicken?
Oh, it's via the feathers themselves. So it replicates in the epithelium of the birds. So like in the actual epithelium of the feather follicle itself, and then that's where the virions are dispersed. So it can live in like feathers and dust and like fomites for months at a time. It's extremely infectious. Yeah.
It's funny. A couple of months ago when you were on, Angela, someone wrote in and said, you need to get a veterinarian on when you talk about these things. Really? Did you listen? Maybe you didn't listen. So this is perfect for this because we wouldn't have known any of this. Thank you.
And what's good is that there's no vertical transmission. So like if you stamp out a flock of birds, if they're infected with Marek's, you don't have to be worried about. I mean, if there were any eggs, it won't be transmitted to the eggs or anything.
So it's chicken to chicken.
Chicken to chicken, exactly.
Okay. Now, so this paper is about trying to understand the increase in virulence over the years, why the mortality has risen, okay? And they talk about virulence evolution in the second paragraph. And I just want to say ahead of time, I don't believe in virulence evolution. I believe it's all about fitness evolution, that there's a selection for increased fitness. And as a corollary, sometimes you have changes in virulence, okay? And for some reason, well, that's something I only recognized a few years ago, I think. But the authors never address this. They make it sound as if virulence is the selection force, right? And we'll get into
Vincent,
that.
Can I rephrase what you just said? Because you said, I don't believe.
Yeah,
Use
you're right.
that word,
Give me another word.
believe. Well, that's okay. You're accusing these people of using a wrong word also. So
yeah, not belief. You're right.
So
I do not agree
how much evidence would you need to convince you that there is a selection for virulence? What kind of evidence?
No, I wouldn't look for a selection for virulence. Virulence is increasing. I'm not arguing that virulence is not increasing. It's clear.
Okay.
But it's not what is being selected for, right? Evolution you select for some property. And selecting for increased virulence is absolutely makes no sense whatsoever. However, increased fitness is easily selected for. If you have an unfit virus, for example, the Sabin poliovirus vaccine strains are unfit. And as soon as they go in the gut, they replicate. And within a few days, more fit viruses are selected for. And they have increased virulence because now they replicate the higher titers and they can get in the CNS, right? So the virulence is a corollary of increased fitness. And that's what I think is going on here. But I think you would need to prove that whatever mutation you think is responsible for increased virulence, which is what they talk about here, is actually improving fitness. So you need to have an assay for fitness, which is often just replication, right?
Well, no,
What
I
if
think
it doesn't?
I think that what you'd have to have is you'd have to show that it didn't impact any other aspect of fitness other than virulence. That would be what would convince you that it was actually a selection for virulence.
Right. Right.
But I have a hard time imagining how you would.
Yes,
be
that's
able
a
to
good way to put it.
figure
In
that out.
fact, but nobody looks for improved fitness. Here they're looking at, as we'll see. But I just wanted to say, because they're talking about virulence evolution, I just want to make sure you understand that it's most likely a secondary effect from the real selection, which is fitness evolution. Okay.
Right.
So the cool thing about this paper is, since we know that the mortality of Marek's in chickens has increased over the years, can we look at genomes of the virus and make some conclusions? And so they look at some ancient viruses, virus isolated from skeletal remains of archaeological chickens. Archaeological chickens.
I'd never thought of an archaeological chicken until I read this.
I love
Isn't that
it.
funny? Can you buy it in the supermarket? Archaeological chicken? It's a good name. So they got a number of ancient chickens, and they
had almost 1,000. No, it was like 995.
995 archaeological chicken samples from 140 Western Eurasian archaeological sites. And they sequenced nucleic acids from them and identified the ones that had Marek herpes virus sequences in them. And then, as Kathy said, they sequenced DNA from the feather of a modern silky chicken that was Marek's infected. And then they had an ancient sample that had no evidence of Marek infection after sequencing it, right?
Well, they had many sites from which they got these archaeological chickens. It seemed like the main one that they focused on was the one from Buda Castle. And because I had been in Budapest, I went on this long thing. It took me a really long time to read this paper because I kept going back and looking at maps of Budapest and remembering places I'd been, and all about the castle and the parliament building and the shoe exhibit on the side of the Danube and all kinds of stuff. But this particular chicken was featured a lot.
So this is not a long period of time. I hate to tell you. In archaeological terms, 1,000 years is nothing.
1,000 years is not. No.
It's nothing. So how far back do you think they could look and still find a virus of any kind, by the way?
Skeletal remains. That's the limitation. Right?
But I think we know about herpes virus. Don't we know about herpes virus from like hundreds of thousands of years ago, Vincent?
Yeah.
We've talked about that.
How about amber?
One of the oldest.
Yeah, you can, and then you can do molecular clocks, and say the herpes viruses were around in the dinosaur era.
Yeah, but
But Dixon, the key here is that
1,000.
So basically, they find that there's a Marek's positive sample, which is 10th to 12th century chicken from eastern France, and so these viruses have been circulating for 1,000 years, and it was not virulent then. So that gives you the opportunity now to compare genomes from then and now, right?
So
because before we only had genomes from after this switch to
Yeah,
the more
that's right.
virulent virus.
I'm going to ask another question though. How long have we been domesticating chickens?
I was thinking the same. I think over 1,000 years for sure, though. No, because it
Yeah.
would have been like BCE. They had at least coops and things like that, not to the
'Cause-
same level that we are now, but
Well,
I mean,
that's the point.
we
That
talk
is the
about
point I am raising.
domestication in general being 10,000 years ago, but I don't know if that was chicken specific.
Right. Well, some dogs and cats were even prior to that, but the domestic animals that you eat or that you use for labor, I think that we need to look further back in history, don't you? That is, we need an explanation as
Googling.
to if you put
within the last
tons
I'd say
of chickens together.
Googling
within the
says
last like
8,000
years.
years ago for
What was?
genomics. Googling says
Oh.
genomic studies estimate that chicken was domesticated 8,000 years ago in
8,000.
Southeast Asia and spread to China and India 2,000 to 3,000 years later.
Right. So that's where I'd
8,000.
look for my bones.
Well, we don't
And
need
old
to.
bones,
The point is we got enough for this exercise here. Okay, I understand what you're saying.
Because the virulence increase really only started within the 20th century.
Yeah, they kind of talk about 1907 versus
1920s.
1920s.
Yeah.
Which is also when like industrial farming kind of started spiking as well, and chickens would have been in these thousands of chickens in an aviary, which you wouldn't have seen before, maybe.
Exactly. When was Frank Perdue born?
Okay.
Google.
So they have a lot of Marek's disease genomes from then and now, and they do phylogenetic trees, of course, and they show that Eurasian and so these ancient sequences, the Marek's disease virus sequences, are clearly ancestors of all the modern sequences. You can see from the phylogenetic trees. Okay, that we say that they're basal to all the contemporary sequences. And interestingly, the Eurasian and North American viruses have distinct clades on the phylogenetic tree, which they say indicates there's been very little transatlantic exchange of the virus.
Yeah, they mentioned somewhere, again, a thing I would not have ever thought about if they hadn't mentioned it, that a bird that was positive couldn't possibly make it across the Atlantic.
That's right.
And that's what made me start to think about...
on a boat. It would die on the boat on the way over, or on a plane.
That was part of why I wanted to know about transmission.
So Angela, are chickens shipped from Europe to the U.S. and back? Do you know?
They would have been before. Now, no. Now that's not common practice because, like, for the OIE controls the transport of animals in, like, the United States and Canada and vice versa. There's many. You don't want to be crossing, like, exchanging animals if necessary, just for, like, different diseases, pathogens. Right. So maybe before, though, 100 years ago, I could see that definitely happening. But nowadays, unless you're trying to import some specific like breed of chicken, it's a specific, I don't know, like hybrid chicken that you want for something, but like just your broilers and your laying hens and things like that, those don't really travel anyway. They don't really live long enough to anyway. They only live a couple of weeks, like the broilers, the ones that are used for meat consumption, like poultry. And then laying
other birds
hens
besides chickens get Marek?
So there's turkeys as well, but it's a different form. It's a different alpha herpes virus. It's a turkey-specific one.
So migrating birds of various species wouldn't be suspect for carrying this virus from chicken to chicken.
Well, so I don't know, but as far as I know, the Gallid alpha herpes viruses only infect chickens, and the turkey virus doesn't infect chickens, and vice versa. So it's quite like species specific. But they can use the turkey virus to vaccinate chickens. But apart from turkeys and like gallids, like galliniforms, like chickens, I'm not actually sure.
I mean, this is a big disease among non-edible chickens also. I must tell you that fly fishing is a big industry worldwide, and the chicken feathers of various specific kinds, Andalusian blue, for instance, or grizzly, and if you go to Southeast Asia, you can see the origin of chickens It's from a jungle cock, which has absolutely stunningly beautiful feathers, which is why they nearly extincted it to get the feathers for fly fishing. When the Brits moved their colonies around the world, they, I wouldn't say raped and pillaged, but they came very close to it with regards to avian species, which they then shipped back to the mother country for inclusion into brand new versions of salmon flies and trout flies, etc. You can see them. It's almost like looking at a book of extinction when you look at these books of all those feathers that they've used.
And they probably brought a lot of Mareks in those feathers. If they
That's
didn't
true.
know,
That's true.
because
So
it would have been in the.
eventually they wised up and now they ship the eggs in, but those eggs are shipped and then they're hatched in certain areas. But you say that's not a way of transmitting the disease.
Not in
That's
the
good
egg
to know.
itself,
That's
no,
very
it's not
good to know.
vertical. Which is probably why this strategy is much more beneficial than
Right.
sending the actual feathers full of Mareks.
So you can't.
You could probably use some of those same samples you're talking about, Dixon, to potentially get more virus samples.
So you can't transmit this over and over and over, so to speak.
No.
Bad
All
pun.
right. So we
Angela
have
didn't get it.
Eurasian and North American strains, distinct clades. They don't exchange much, right? But they have both become more virulent over the years. So they have independently become more virulent. All right. So that's it. That's an interesting observation. So they look at the genome changes to try and get an idea of what might have caused this increase in mortality. And they say there are no major gene losses or gains. So probably there's a point mutation, and they have 158 fixed single nucleotide polymorphisms between the ancient and the modern samples. And some of those are in between genes, so they don't talk about them here. But 49 open reading frames with evidence for positive selection. In other words, an amino acid change occurs and then it remains present in all the viruses after that. Because if it didn't matter, well, first of all, if it's lethal, you would never see it probably because the virus wouldn't exist. If it's neutral, it would probably be lost at some point. But if it's positively selected for, in other words, it confers some advantage to the virus, which I would say would be fitness, it remains. So they have 49 open reading frames. They go through a few of them and talk about, there's ICP4 is a protein that's an antagonist of immune responses. So obviously changes in it could affect virulence. There's some viral glycoproteins, which are involved in immune responses. There's an interesting protein called a viral chemokine. It's a homolog of a cell chemokine, so it interferes with the activity of the cell chemokine. It's called VIL8. And so all of these are candidates. There are changes in these, and all of those are candidates for changes in mortality. But they don't go further. They look at a gene called MEQ, MEQ, which is a transcription factor that regulates tumor formation in Marek's disease virus-infected chickens. And so, but remember, it's a transcription factor, so it can also affect other genes as well besides tumor formation. And they find a number of changes in the MEQ gene. And in particular, they focus on the C-terminal domain of the protein, which is important for this tumor induction. And they find tetraproline repeats, right? 4 prolines in a row, and this is repeated multiple times. And what they find over time from the ancient to the modern is a loss of tetraproline repeats. And they think that this is what's happened from ancient to the current, is that you lose these tetraprolines, and they're important. The loss of those then increases the transcription of the tumor-inducing gene. Okay, so they say loss of tetraproline motifs acts as a ratchet in which each subsequent loss results in an increase in virulence. And once you lose these motifs, they can't be regained. So there's where I object. It doesn't, you get an increase in virulence, but probably the primary effect is an increase in fitness of the virus, which they don't mention. I think they should mention that. And as I said before, an increase in fitness, and then that causes an increase in virulence. The word fitness is mentioned once in this paper, right in this area where they're talking about the tetraproline receipts. They say, repeats, let's see, local fitness peaks are outcompeted by lineages following the order described here. So they kind of get close to fitness, talking about fitness. So I think that's what's happening here. The loss of these tetraproline is somehow increasing the fitness of the virus. We don't know why, because nobody has studied it. But then a corollary is that same protein makes tumors, and so it increases the virulence.
Right.
So the same.
And those two things could be really closely related or not. So maybe this transcription factor changes transcription of some random viral protein and also happens to change transcription in a way that leads to tumors. Or maybe this protein is impacting cell cycle to make the cell's environment more favorable for viral replication. And that exact same cell cycle change also increases oncogenesis. And so I think the point is that the goal, not that viruses have goals, but the goal for the virus is not to kill chickens. The goal is to replicate better. And it just so happens that the thing that makes the virus replicate better also leads to killing more chickens as like a side thing.
Yes, this is exactly right. And I do think they should have discussed it. And if you ask the authors, they will say, well, they didn't let us add any more words. They will use that as an excuse. Absolutely. I guarantee it. But you know what I would say? So take some other words out. You don't need them all, because it's really important what Brienne just said.
Right. But don't take out the word ratchet, because I know that Vincent and I really enjoyed seeing
ratchet's
that.
A good word.
Muller's ratchet. Cool explanation. Yeah.
So they actually do an experiment now, which is good. They say, okay, let's take this MEK protein and see if it has any difference, if the ancient MEK and the modern MEK have any difference in driving transformation. So they introduce it in a cell in culture, and they have a promoter that's responsive to MEK, and the promoter is driving, what is it, a luciferase, I think, right? Something easily assayable. And the contemporary MEK protein, which is from very virulent viruses, transactivates, we call it, this promoter 7.5 to 10 times more than the ancient one. So it's consistent with the idea that the changes over time in this MEK gene are responsible for increased tumorigenesis and lethality. But as I would say, and Brienne would agree, it's something else probably too that they're not looking at.
Yeah, it would be really cool to do this exact same thing and see what other kinds of changes happen in the cell with ancient versus contemporary MEK.
Yeah, exactly.
And just to make it really clear, they don't put the protein in directly. They do molecular biology. They put the gene in for the MEK, the ancient MEK, and they compare it to the modern MEK, which I think they also synthesize that from scratch.
Yep.
Right. So almost all this study has been done in domesticated chickens.
Yes.
So I'm going to ask a different question, though, because, whether or not the virus is more or less fit to replicate inside of a single species, which is specific for, what if it kills all of those animals and makes room for something else? So on an evolutionary scale, this virus still plays a very important role in making room for something that ordinarily wouldn't have been able to take over the habitat of this wild fowl, which later on became domesticated. And population genetics plays a role here too in balancing out populations of various species by their infections. And I don't see any problem with virulence as a selection, not for that particular virus, but for another life form that takes advantage of the fact that it leaves a vacuum that it can now fill.
So I think that's a really interesting kind of population ecology type of thing. But when I think about sort of molecular mechanisms of how exactly the changes are going to feed back on making a virus more fit, I have a harder time imagining how does having different biodiversity of organisms change which amino acid in the virus is more fit. I don't know if I'm phrasing that right.
Yeah,
but
I understand. All right, so that's the paper. Basically, the idea is that ancient Marek's disease virus didn't cause tumors. It infected the chickens. And they say here, this is very interesting, characterized by slower viral replication, low levels of shedding, and lower clinical pathology. That's a less fit virus. That's the characteristic of a less fit virus, lower viral replication. So over time, you select for higher replication, and as a consequence, that selection led to tumor formation. And that's the way I would have phrased it, but they don't. They never go there. I don't know why. I don't know why. You know, in science, we have different interpretations, right?
Right.
But in this case, fitness is the right one. There's no question about it.
Right.
Okay, that's our first paper. Very interesting. And actually, Steph Langle sent it to me last week, and also Amy. So thank you all for sending it. Second paper, Nature Communications. This is selected in honor of Dixon de Pommier. This could be a TWiP paper, but the hell with that. Yeah, we are doing TWiP today, but we're doing this paper on TWiV, and
This is true.
because I want to introduce to the TWiV audience this virus. So it's a Nature Communications, Diversity and Dissemination of Viruses in Pathogenic Protozoa. So here we have, none of my highlighting is on this copy. Why is that? That's going to be tough. Oh, no, I'm not on my highlighting. I'm going to go to another computer because I cannot live without highlighting.
While you do that, I can tell you that it doesn't appear that there's any shared authorship, but the first author is Serena Heron, and the final author is Frederick van den Broek. And I'm
also talking about Leishmania does not affect chickens. In case anyone
right, of all these authors, I only recognize Stephen M. Beverly. I was at one point a very close friend of his, and we used to meet at Gordon conferences all the time and discuss Pat Metheny records, because he was I presume he still is a big Pat Metheny fan. But he worked at Harvard. He got tenure. He had his own big laboratory, and then he moved from there to St. Louis, where he still is, I believe, with his wife, and she was working on muscle diseases. So we had a wonderful set of circular conversations about viruses and Leishmania, and then mussels, and then Trichinella, and then the whole thing got together over Pat Metheny records. So I think that
look at Dixon dating himself. He says records.
Well, we had we had some CDs at that point. We did, but Steve was a very, very intense, and still is very intense, and very committed to his subject, and widely respected among everybody who works on leishmaniasis. So when his
Dixon,
name is on it.
Is Leishmania or Leishmania?
Well, Leishman is the name of the guy.
Okay.
And
so we'll do Leishman.
I think Leishmaniasis.
All right. Did you do the affiliations of these authors? I don't remember.
I only read the first and the last, and did not read their affiliations. But Antwerp, Belgium, whatever.
Okay.
So it's open access also.
Leishmania are unicellular protozoans, and they can be infected with viruses. In particular, they're infected with double-stranded RNA viruses. The family is called Totiviridae.
Right.
And one interesting aspect, there are several, there's no lytic phase of these viruses. They persist in the cell. They don't kill the cell, and they just go from cell to cell when the cell divides.
Right.
So Dixon, I have a basic question.
Sure.
About Leishmania. So, we have a picture in the show notes of a replication cycle that you have that's come from you. Are these parasites obligate parasites, or can they be free living? Because I see them here in a sandfly or in a reservoir host or things like that. But can they also just live by themselves in the environment?
They can be grown in culture. So you could intimate the promastigote stage can actually be grown in culture. apart from any host material of either from the mammalian host or from the insect vector. But it won't progress to another stage by simply culturing it in the goodies that it needs to stay alive. It'll replicate, but it will not transmogrify itself into a flagellate obligate intracellular parasite, which you see on the far right
mastigote, right?
diagram at 9 o'clock. You can see, or let's see, that's 3 o'clock. Sorry, 3 o'clock. You've got what they call an amastigote, which means it doesn't have a flagella. And you have a promastigote, which has got a flagella. And then if you go through the sandfly gut, which you shouldn't do, I don't recommend that to anyone. The sandfly is this innocent looking little absolute wimp of a fly that has a very short flight range and lives near the burrows of animals that burrow at the root systems of certain tropical trees. And one of the trees is the chicle tree. And the chicle you might recognize as a tree that gives its sap up to the chewing gum industry so that we can have chiclets. I'm not joking. And so
that's where the
chicle
name comes from?
gatherers, yeah, that's right. You see, you learn a lot by studying parasites because it's not a standalone subject. They need other things, right? So chicle gatherers are prone to catching this over other occupations in the tropics. And the earlier you get to the tree, of course, the more likely it is you'll be the first one there. So getting there early is a big mistake because the sandfly loves early arrivers, and they come out of their little burrows next to the animal burrows, and they take advantage of the fact that there's someone standing there cutting these V-mark chicle paths for the sap to drip through. So it's a big deal disease. This particular species, and there are as many species of Leishmania as there are countries in Central and South America. Each one has a slightly different genetics. They're all basically related to each other. The Leishmania braziliensis is distinctly different, and it's different in a very sinister way. It looks exactly like the other species of Leishmania, except when it enters the mammalian host. It starts off with a lesion that's recognizable as a Leishmania lesion. You sample the margin of the lesion, you find the organism, you say, aha, and you find the organism by putting it into culture. And the culture is in a medium which encourages the growth of insect tissues. So the culture is called Schneider's medium, and Schneider's medium is used to raise insect cells of various kinds, like mosquito cells and that sort of thing. And this promastigote responds well to that environment because that's where it's heading. The mosquito picks up the amastigote stages, and miraculously, they rapidly transform after their excess material is digested away from them. The amastigote stages, the one without the flagellum, starts to grow a flagellum, and it becomes part of the internal intestinal life cycle of this parasite, which eventually ends up penetrating into the hemocoel of the Leucemia in this case. That's the genus name for this sandfly, and enters the biting mouthparts. Now, when the insect bites another individual, it had to bite one to pick it up. Now it has to bite another one to transmit it. It injects the promastigotes into the site where it's deriving its blood meal. And once it withdraws its mouth parts, this promastigote is eaten by a macrophage. Ha ha, gotcha! And that's really not what happens. It's the promastigote that wins because this little organism has the ability to penetrate out of the vacuole that the macrophage creates, the phagocytic vacuole, and penetrates the membrane of that vacuole and enters the naked cytoplasm of the cell. And at that point, it becomes unrecognizable to the cell. It's like it was another organelle, but it's not, of course. It's a parasite. And the
How is
parasite
it possible
begins.
that it doesn't set off all these innate like sensors,
Yeah,
and how
it
is it?
happens
How?
real quick.
Okay, but the
There's no time
macrophage
for any
dies.
of this.
But these mastigotes lyse the macrophage. That part I
No.
actually don't know.
Eventually
Or
they do, of course.
they do eventually, though, right? It is
Yes,
like
they
a
do. It's like
lytic.
a virus
Okay.
at that point. It
Yeah,
is like a
okay.
virus at that point. It multiplies inside the cytoplasm of the macrophage, and when the macrophage can no longer biochemically or physiologically or ecologically support the growth of more, it's finite and it bursts open, and then these amastigotes are transferred to the next phagocytic cell, and that continues. And this organism thumbs its nose at lysosomes. It has no interest in lysosomes whatsoever, so you can't kill it by the typical pathways that most pathogens are killed by. And that's why it's so sinister, because once it gains entrance into a wandering macrophage, then it can be transported to any part of the body. And unfortunately, oftentimes, it ends up in the nasopharyngeal region of the mouth, of the oral cavity, I should say. And it starts a new set of infections, which eventually you don't want to see the pictures of people suffering from this version of it. It's called Aspundia, and it's a horrible erosion of the entire soft palate inside of your oral cavity, including the nasopharynx.
It must be able to infect monocytes, no? Because if they're just tissue-resident macrophages, those wouldn't be traveling to the nasal epithelium.
That's right.
Like those wouldn't get there. They would have to be in monocytes that then differentiate into macrophages, no?
Sure, they can infect a variety of phagocytic cells, and including the neutrophils, but in dendritic cells, it can infect them as well. And in fact, that's probably the first line of infection that they encounter when they enter the skin. Would be not a macrophage, but per se, but rather a dendritic cell. Nonetheless, the phagocytic pathway for protection, which I guess we would call that the second line of protection, fails miserably with regards to Leishmania. And you end up, and then there's another one like that too, called Chagas disease, which it's related, but not directly related to this particular protozoan. And that too has the ability to sneak into the heart muscle cells and disappear from the radar screen. And what you've got here is a stealth parasite that eventually the lesion on the skin disappears. The clinician says, "Oh, thank goodness you're cured." This has a certain length of time it takes for this curing to take place, and then 3 to 5 months later, you start to see this erosion of the soft palate. And occasionally, and I'm told the urogenital drawing here is probably not representative of the typical pathway that this organism takes. You could probably put an X through the urogenital mucocutaneous membranes as a source of infection. There are very few reported infections in the literature, but the typical pathway is the oral pathway.
So I was the one who put this figure in because I have never downloaded parasitic diseases, and so I wanted to do that. For anybody who doesn't know, you can go to Parasites Without Borders and download this. Yep. And then, because I kind of wanted to have the bigger picture of where protozoans are with the other kinds of parasites.
Good idea.
So I guess the reason why I asked that question is one of those interesting things that comes from us thinking about two different papers on the same day, and maybe a thought that I wouldn't have had if I hadn't previously thought about the other paper. So here we're going to be thinking about a virus that infects this protozoa.
That's right.
And so, part of me wondered, well, what does the virus just do to the protozoa, outside of when the protozoa is in the host? But it sounds like by and large, the protozoa is almost always in a host, while yes, you can grow it, as you say, outside of a host in the culture. In terms of sort of biologically, when we actually see the full life cycle of this protozoa, it's happening in a host. And so here, doesn't make as much sense to think about the virus and the protist and not another host. Here we need that other host as well.
We do. So I should also say that there are other protozoans that have double-stranded RNA viruses associated with them, and Trichomonas vaginalis is one of them. Another one is Giardia lamblia. These are common protozoan infections in people. And so the authors wonder whether or not there is an association of virulence of the protozoan in humans that have or don't have viruses associated with them. And they go through some really complicated population genetics, I would say, way over my pay scale. I'm glad you didn't ask me to review this paper.
You may remember many years ago, Dixon, on TWiV, we talked about a paper where they showed that these double-stranded RNA viruses, Leishmania RNA virus, for example, it's sensed in the mammalian host by toll-like receptor 3. Right. This causes a hyperinflammatory response. It increases disease pathology because there are some Leishmania without RNA viruses, and the pathology is in general reduced compared to the ones with RNA viruses.
Yeah. You know, it sounds a little like the story with Wolbachia with worms. There's an association going on here someplace. And that, by the way, that parasite was not Leishmania. I believe we were discussing Trichomonas vaginalis.
May have been Trichomonas, yeah.
Yeah.
So and also from within the amastigote, it's sensing, how does TLR3 sense this virus if it's inside of the amastigote, inside of the macrophage? Or is it only when those amastigotes lyse or somehow
I think they have to lyse, yeah.
Or they're like, okay.
They have to lyse because the RNA has to be sensed.
This thing, they've actually used molecular tracers, the fluorescent tracers, in the promastigote to show the events that occur in cell culture with macrophages and how fast this organism goes from the outside to the cytoplasm of the macrophage. And it's a matter of seconds. It's not a slow process. This happens incredibly fast. So there's you know, they came and they went, and you don't even know they were there.
The other aspect that's interesting, Dixon, is that what are these treated with? What drugs?
Well, they have some interesting drugs. Let's see. Let me think about this while you surprised me with that question. We could go to the book and look it up.
Oh, it's okay. But anyway, the drugs that are used, they often fail, and when they fail, they're often with Leishmania with viruses in them. So the viruses are associated with increased pathology and drug treatment failure. That's what I wanted to point.
Right.
So in this paper, they wanted to say, is there any relationship between the host and the virus that we could pick up just by doing genome sequencing? So they get isolates of Leishmania braziliensis from different places, and then they sequence their genome and that of the associated Leishmania virus and try and make some conclusions, which will muddle our way through. But basically, they have gotten samples from, Brazil, Peru, and Bolivia, 79 L. braziliensis isolate, and they know whether or not they have Leishmania virus in them. And then they sequence the genome of the Leishmania, and they sequence the genome of the RNA virus as well. And one of the things they note is that when you build phylogenetic trees of different sorts on these the Leishmania genomes, they say, looks like there's a lot of recombination going on among these different isolates, right? So there's a pattern. Long branches on trees are symptomatic of recombination, which is quite interesting. They say, well, maybe this is a prevalent thing in this species, right? And it just hasn't been studied all that much, so it's not clear. But despite that, they do find some isolates with almost identical genome sequences. And they think that these represent clonal population in the wild somehow, and they're geographically confined, yet they can see them circulating for 12 years or so. So almost identical host genomes, right? And as compared to thousands of differences in the genomes of other viruses. So I thought that.
So Vincent, let me just interrupt by answering your older question. Now that I could go to the literature, because there are new drugs out for this organism, and the ones that I was going to mention are no longer being used. Miltefosine is the drug of choice for visceral and for cutaneous leishmaniasis.
Okay.
But ketamine No, not ketamine. That's not what I'm going to say.
No, that's not ketamine.
No, it's not. It's
In dogs,
the
you can.
antifungal drug. There's an antifungal drug.
It's one of the azoles, probably something that
Yes,
ends in azole.
that's right.
Yeah.
That's right. You can use that to treat topically for cutaneous leishmaniasis.
You mean like metronidazole?
No, there's another one that's used, ketoconazole. Ketoconazole is the one I was thinking of, and that's the one that's very effective against cutaneous. And when you treat these patients with ketoconazole, Leishmania braziliensis, it simply exacerbates the mucocutaneous aspect of it by saying you can make it disappear from the skin, but you can't treat the systemic infection.
Okay. All right. So we have highly related Leishmania braziliensis, but they also find some groups with mixed ancestry, which they call hybrid groups, and those are important. They're going to have a little bit different properties, as we'll see. They also look at the effect of geography and the environment on the populations, right? They try and make, these are all associations, statistical association, where they make models and say, could geography, climate, right, dry months and wet months, could that affect the kind of Leishmania that's circulating? And, one model, environment and geography explains 27% of genome variability. In another model, it's slightly higher. So you can associate some of this variability. In other words, the changes may be responding to climate and environmental changes and so forth. But the three hybrid groups that I mentioned, these are more widespread than the older parasite populations that are mostly in the tropical rainforest. So somehow this hybrid aspect of the genome is giving them an ecologically broader range. All right, so that's the host, right? So you have these constant hosts, which are more ancient, and then more contemporary are more hybrid. So then we look at the virus. So 31 out of these 79 Leishmania were positive for the virus, Leishmania RNA virus 1. They get coding, they get almost full-length sequences of many of these viruses. These viruses, by the way, are relatively simple. They encode a capsid protein and an RNA polymerase in overlapping open reading frames. So they're highly dependent, of course, on the host for other functions as well. And when they build phylogenetic trees, they basically can see that the viruses group with specific hosts, right? The virus strains cluster with their Leishmania host species, which is consistent with this idea that they are moving horizontally, right? Yes,
it's important to point out that these parasites are promiscuous beyond belief.
Mm-hmm.
I mean, almost virtually every mammalian species in South America is susceptible to them, including humans, of course. So there's a ton of reservoir hosts. In our life cycle drawing, we didn't have room to put all of the mammals that this organism has been found in. So this organism has enormous opportunities to pick up stuff, as it were, from other hosts, and by encroachment and by replacing forest with farmland, by mining, by urbanization. That's the way this organism has come about.
Yeah, they actually
we
talk
created
about that a
this
lot,
organism.
Dixon, when it infects other hosts, does it cause any pathology?
Absolutely.
Yeah.
Absolutely. Yeah, this is not a benign infection anywhere else. This, you can see the lesions, you can tell who's got it, and visceral leishmaniasis, which is Leishmania donovani, is an incredibly pathogenic organism, which when you catch it, it's very, very difficult to cure you.
Dixon, if you have like cows next to this area, would they get infected?
Cattle
get
it.
They're susceptible to it as well.
Yeah,
they do.
And horses and every domestic species, at least in vet school, we learned mostly cats and dogs. It's very severe in dogs, all three forms of Leishmania.
Exactly right.
But
and pigs,
cattle as well,
also
and pigs. Yeah.
And there's a lot of peri-domestic pig husbandry, and chickens. Not chickens. No, this is
not
mammalian
chickens.
disease.
No, mammalian. Yeah.
Sandfly. It depends on the preference biting habits of the sandfly rather than the parasite. So in this case, the sandfly is promiscuous, and it will take a blood meal from virtually any mammal that it encounters, which is highly unfortunate for us.
All right, moving forward with the viruses, Peru and Bolivia, 9 divergent viral lineages. Okay, different, no recombination, which I'm good. I wouldn't expect to see recombination here, and they get multiple viruses within the same geographic region. So they say actually we have undersampled. There's probably a lot of more lineages than we know of because we've only sampled so many. And most of the lineages were found in the single area, so that they say maybe the spread of this virus is restricted. 2 are more widely distributed, and nearly all lineages were found in the tropical rainforests of Peru and Bolivia, except for one that was in tropical rainforest, tropical monsoon, and tropical savanna and temperate climates. So they say we think the virus evolved mostly in lowland tropical rainforests before spreading to other regions, as Dixon said, by anthropogenic activities. Mostly.
Exactly. And it should also be quickly pointed out, these are just 2 countries in South America that were sampled.
Of course.
Every country except Argentina has endemic infections of this organism. So what's going on in Peru and Bolivia is probably going on all throughout the South American continent.
Right. They also find that these hybrid lineages are have different prevalence of these viruses compared to the non-hybrid. Lower prevalence in of the virus in the ancestral populations, higher prevalence in these 3 hybrid populations. That's the host that I talked to you about. And then they say, what's the association between having that virus and treatment outcome? So they have 41 isolates for patients with known treatment outcomes. 23 patients were cured and treatment failed for 18. Percentage of treatment failures was lower for isolates without the virus. It's than for the positive. So treatment failure, 37% in virus negative, 53% in virus positive.
These were all treated with miltefosine?
I don't know. I didn't pick up.
I don't think they said
that was there. So most of the patients with treatment failures in one of the hybrid groups, so remember that's the Leishmania host itself, were 88% associated with a specific viral lineage. And so in this particular hybrid group of Leishmania, most of the treatment failures had viruses in them. So what do we learn from this? We learn that the host is genetically heterogeneous. It is structured according to geographical origin, but they say we've only captured a little bit of diversity, as Dixon said. There are only 2 countries here. It's all over the place, right?
Right.
Yeah.
But they talk quite a bit about how human activities, environmental destruction, migration of humans, reservoir hosts like dogs and cats have resulted in movement of Leishmania and their viruses out of the rainforest and into other areas, which is what their data suggests is happening here. So that's the story. Dixon,
well,
any comments?
That's the tip of the iceberg of the story.
Yeah, indeed. Well, this is a little study, right? I'm sure it's going to change.
Yeah, where the Trans-Amazonian road, which led from one of the coastal cities to the center of Brazil, so that they could establish Brasilia as the capital of Brazil. They wanted to do that. They built this absolutely failed city in the middle of Brazilian jungle, and in doing so, they created what we would call an ecotone. The ecotone is the ecological region on either side of something else. Let's say a mountain and the fields that come up to the mountain through the foothills. If you draw a line right down through the foothills, the mountainous ecosystem is here, the field ecosystem is here, and that little zone in between the field and the mountains, that's the ecotone. That's the mixing zone for animals that migrate from the field to the mountains or mountains to the fields. And we do the same thing. We're just one of those animals that in Brazil, for instance, there's a workforce that annually migrates from the coastal cities inland to help with farming, mining, and other activities related to labor forces. And then they come back to their cities to spend their winters or whatever they're doing. And the same, by the way, in China, the same thing occurs for different reasons. But we migrate, I think, probably more than any other animal on the planet. And then you can imagine how we can redistribute diseases that are endemic for certain places. Once we catch it, we are happily give it to somebody else and share it with all the other vector
Dixon,
species.
Are you sure we're happy about it?
Yeah, right. Dixon, so if I go to a big city in one of these countries, Peru or Bolivia, for example, am I likely to get bitten by a sandfly?
No, not at all. No,
so you have to be in the countryside, right?
Well, not quite. You could be in the suburbs.
Okay.
You have to have vegetation. You have to have animals that are also present that are not considered pets. And that's why the chicle gatherers, they've earmarked every single chicle tree in their vicinity.
And they get infected, right?
They get infected regularly, absolutely. And that's another thing. There's, I don't know how many different species now of Leishmania, but most of them are cutaneous species. And most of the cutaneous species of Leishmania confer immunity against the other cutaneous species, but not against the visceral or the mucocutaneous varieties of these Leishmania species. They all look alike. There's no way to tell them apart on a microscope. You've got to do the genetics to actually figure them out. Now, I would like to ask a question. In Racaniello-infected Onchocerca volvulus worms that cause blindness, if you cure the Racaniello, you don't get blinded. All right. What happens if we were to cure the virus infection in the amastigote stage? Can we do that?
Yeah, but I think you still get disease, right?
You get disease, but it's a mild form, right? And it's not drug resistant. That's the big advantage. It's not these
so
people.
You want to treat the patients with an antiviral?
Well, I don't know. A double-stranded RNA.
But it's a double-stranded RNA virus that doesn't have an extracellular stage.
Exactly.
Yeah, but it's still it's got an RNA polymerase. We could make drugs against that. The problem, Dixon, is you say that you get a lesion. You get a lesion, and it goes away, and they say, "Oh, you're done," right? So
I know.
I don't know where the antiviral would come in.
Well, I think you'd figure out whether they've got the virus first, right? Because if you give them the drug, if you give them the drug and they've got the virus, it's not going to go away.
Well, it might. Depends what the drug is doing, right? The virus has to reproduce to get from cell to cell. So if you inhibit reproduction, it could work. But
didn't this paper just say that the people that have the virus don't get treated as well?
Yeah, they have drug failure. But I'm just saying
exactly
that's drug against the parasite. But if you had an antiviral against the virus, presumably that would be okay, right?
And then you could use the traditional drug and cure them.
Maybe, yeah. Maybe you could do both at once.
Or maybe you could do both at once.
Yeah,
so you recommend working on a viral, and that's another question, of course.
Well, Dixon, how prevalent is this infection? That's what's going to drive making antiviral.
You know that this is a poor person's disease in a poor person's country, where drug trafficking is the biggest way of maintaining employment. At least in Bolivia, it was when I was there. My wife and I took a trip to Bolivia and had a wonderful time, but we learned that over 60% of the people in Bolivia are employed by the drug cartel.
Yep.
60%.
Question, Dixon. These chicle gatherers, these people that are at these at these trees. I mean, they're not 100% going to get infected, but it's extremely high chance of them getting infected. So do you know if there's any sort of like preventative measure that they take or that they can
there is.
take?
Here it is. Leave after 10 o'clock in the morning to gather your chicle and you won't catch it.
So before 10, so they come out in the morning like
That's
first.
correct. That is absolutely correct. So
as opposed to any sort of
drug. Don't stay late. But if they enacted a law that says you can't gather chicle until 10 o'clock, then they would say, well, then there goes my livelihood because someone else is going to beat me there, or it really interrupts my work
Yeah.
schedule, and it's a difficult thing to
I
actually.
suspect that law enforcement is not easy there, Dixon.
Yeah, well, I think we should ask Daniel Griffin that question. He's been into a lot of those countries, and it's a slippery slope everywhere you look.
Yeah, he's been in a lot of places. He said he's been bitten left and right, and
He's been into
fine.
places that I would never go to.
He's fine. He never got anything.
I would. Oh, that's not true. He's come back with diarrheal disease and things like this.
Yeah, he told us a story on TWiV 1000 about
that's right.
He got dengue, yeah.
And that's another one.
bad.
That's right. But he's dodged bullets. I agree with you, and he's careful. He says he's careful, but I think
he
a lot
towels
of these
off rigorously. Yeah,
these other people are careful too.
So there was a TWiP called toweling off rigorously. He said he used to swim. He used to swim in like Lake Malawi, what's in the lake that can get you?
Schistosomiasis.
So he says he comes out and he towels off vigorously to get rid of the parasites
that's what you should do. And
is that right? Because otherwise they go into your hair follicles. Is that right?
Yeah, that's true. That's exactly right.
Okay, let's do a couple of email. That was lovely. Even though the papers are hard, I appreciate the discussion. Thank you very much, Angela and Dixon.
No, I loved it. And thank you, Kathy, for posting that life cycle. That's very helpful.
When I had to jump in was when you were talking about the mucocutaneous lesion, because I got stuck on that page and I thought, it would be horrible to have to edit this page and have this picture there.
Exactly right. Exactly right. Well, we're editing ourselves using Grammarly, so I think we're doing okay.
Dixon, can you take the first email?
Be happy to. Charmaine, of course, Charmaine writes, the beginning of this article mentions a virus in quotes, which is incorrect. Later, the article states that it's caused by a prion, which is correct, and I don't know the author's bona fides. However, I am concerned about prions, even though they're not viruses. Can you guys do a show on prion diseases? I'm about to give up on eating mammals completely due to this.
Oh my God.
I wouldn't do that, but sure, I would be.
So, more and more deer are turning up positive for chronic wasting disease in more and more states, and the press goes nuts. Scientists express concerns as zombie deer disease detected in Yellowstone. Blah blah. So far, and we did an episode a long time ago on CWD and deer, and I got a letter from a lady at some U.S. government agency, and she said, no, there's no evidence that it gets into people. You shouldn't scare people. So you should talk to the press.
Exactly. So, Vincent, am I pronouncing that incorrectly? Is it prion or prion?
Prion. I mean, it's a made-up word. Prion.
You use prion? I like prion.
You read Stanley Krusner's autobiography. He tells you how he came up with this word. He says, I went to the cafeteria at UCSF Hospital, and I was having a tuna fish sandwich when I thought, infectious protein, that's what it is, but INPRI doesn't sound right. So he reversed it, the prion. He was eating a tuna fish sandwich, though. That's the key.
That is key.
Prion. So I think prion, that's what I usually say, prion, but you could say prion.
Okay.
Whatever Stanley said, he's the one who invented it, so he could.
Right.
So we did a lot of TWiVs on prion diseases. The most recent one is 950, which I did in Manchester.
That's not such recent anymore.
Yeah,
2022.
2022.
With Richard Knight, who's an expert on prion diseases. Yeah, and then there are a bunch of others as well.
Yeah, so all you have to do is Google TWiV and prion, and you get them all.
Yeah.
Bingo.
Kathy, can you take the next one?
Yes. Perry writes, Hi, TWiV. You were curious on when the crossover of seroconversion of EBV in a population. Here's one study of the UK population. EBV seropositivity increased more rapidly in females than males during adolescence, age 10 to 15. So somewhere in primary school would be ideal for an EBV vaccine, assuming it will prevent EBV latency. Thank you. So, and includes a link from BMC Public Health.
Yep, epidemic of EBV in UK. Thank you very much. Appreciate that. Okay, what do we have here? Oh, Angela, take the next one, please.
Okay, David writes, Hi, TWiVers. Regarding TWiV 1073's discussion on the possibility of transmitting viruses by carnivores eating infected prey, cats, including domestic cats, are efficient predators and eat many rodents and birds, so lots of opportunities for transmission. The classic case is the microbe toxoplasmosis, or toxoplasma, Gandhi probably, they're referring to, which has obligate feline and prey phases in its life cycle. Toxo even infects the brain and alters prey behavior to make infected animals more susceptible to feline predation. Speaking of brain-infecting microbes that alter behavior, rabies is, in a sense, an example of reverse carnivore transmission. The virus infects the brain and induces hyperaggression and is transmitted to bitten prey. It would be interesting to look at a feline virus, or to look at feline viruses to see how many of them are also found in rodent populations. My guess is that there'll be an overlap suggestion transmission to carnivores. Lots of worm infections are transmitted by carnivorism too. Dixon de Pommier. Did you add that at the end, Dixon, or
Yes.
are they okay? But it's like within the email. Okay, Happy New Year to all, David. I was unclear. I was like, are they referring to Dixon?
Dixon
Okay,
didn't. Yeah.
Otherwise you wouldn't have read it.
I think that it would be interesting to see. I don't actually know, like off the top of my head, to think of how many feline viruses can be found in rodents. I mean, like feline viruses, different coronaviruses, they're pretty specific to cats, like those that infect cats. So maybe like those wouldn't be in rodents. Bacteria, I know there are quite a few. Like cats can get infected with Y. pestis, and like so can rodents. That's a huge like reservoir. But viruses like parainfluenza virus that infects cats, or caliciviruses that infects cats, I have no idea if those are in rodents.
Okay.
It's possible. Interesting thoughts.
Okay.
But
yeah. So someone had asked us last. I think Dixon, you asked the question: how many viruses are So there you go.
I did.
The rabies example is interesting. Obviously, that happens in non-human rabies infections because we don't bite. We don't bite other animals.
No.
Yeah, we're pretty much.
Well, and a lot of it is yeah. Well, humans also get aggressive, though, right? Humans can also have like phases of aggression in rabies, but that's like very advanced rabies, which ideally humans would not have. But
they also get aggression without rabies too. You know,
this is all true.
It's true.
True. Much more probably.
Yes. So we have 2 emails from 2 different people, and they should be read together. Brianne, why don't you take those 2?
All right. Susan writes, I'd love to hear one of your experts discuss the recent dog illness of unknown origin, but likely viral. Colorado State is apparently on the forefront. I've attached an MSM article, Fear-mongering at its best, and we have a link from USA Today. I love the show and the care and concern you put into all you do. Your passion, Vincent, is truly infectious. Justin also writes and gives a link about this that says, novel dog virus. My 8-month-old pup likely had it, definitely nasty.
All right, Angela, you know anything about this?
I think, people like sent it to me as well. It's kind of unclear first if it's a viral or bacterial pathogen, because those that go on antibiotics are like, oh, the animal improves. And it's like very anecdotal up until now. There's not really any sort of, there hasn't been any sequencing as far as I know, or anything to actually see like what this pathogen is.
Yeah, somebody had asked me about it right when a lot of these cases started, and the data that I found, or the information that I found, seemed to indicate it was maybe like a mycoplasma, maybe like a previously unknown mycoplasma or something, and so, it was always super unclear to me exactly what was up.
Yeah, because there's like your typical kennel cough, or all of these agents that cause respiratory illness in dogs that include, coronaviruses and adenoviruses and parainfluenza viruses, even herpes viruses. Like maybe it could just be a new variant, a new serotype of one of those that is escaping the vaccine, because obviously your dog would be vaccinated against most of these for the kennel cough and all when they're puppies, all of these things, and then they get their yearly boosters. It could just be like some sort of escape variant. We have no idea to any of those viruses, if it is viral, but we also don't know. So unclear. And it seems it doesn't really seem lethal. As from what I read, it says 1 or 2 animals had died. It doesn't seem like some sort of fulminant virus that's killing all of its like the infected host. But yeah, TBD. We don't really have that much information about it yet.
We'll see. Next year we're going to learn. I bet.
Yeah.
All right, let me take the last two. Ken writes, lab leaks and biosafety frequently come up on TWiV. The impression I got after hearing this nearly 2-hour-long interview with the author of the book, Pandora's Gamble, Lab Leaks, Pandemics, and a World at Risk, gives a link, is the risk much greater than the impression I've gotten from TWiV over the years. The author had a short piece in The Guardian touching on the issue. The benefits of research that requires biosafety is many and important, but I now wonder if the TWiV crew has been downplaying the risk side of this. Maybe the message is only that better safety practices are needed, or maybe sometimes the risks are too high. Ken, why would we downplay the risks? What horse do we have in this race? We are virologists. We understand the benefits and the risks. We don't have a company that's doing this, so why would we downplay it? Doesn't make any sense to me. And in fact,
we also don't particularly want to get hurt ourselves.
No, we want to be safe. We want to work safely. And everyone I know who does BSL-3, BSL-4 work, works very safely, is very concerned, follows the protocols. This business about hidden lab leaks, I don't know about any of that. I've never heard of it, and I don't see why a journalist would learn anything. It's, as far as I'm concerned, nothing serious has ever happened out of a lab. Yeah, you get little things. Ron Fouché was on TWiV years ago. He said, I have a computer in the BSL-3. When it crashes, I have to make a report. That's how comprehensive the monitoring is. So I think the safety is really good. If you've been in a 3 or a 4, you'll see that.
Yeah.
Go on.
Everything takes 5 times as long because there's so much safety. Everything takes so long.
It's because people don't know how it works that they're suspicious. Go look at the BSL-4 documentary, The Threading the Needle, that we made. I think it's pretty telling how everything is triple redundancy, safety left and right. Elke Mühlberger, who was on that episode, said she was pregnant and she worked in the BSL-4. She felt safer than she was in the real world. So we do not underplay the risk. We are realistic, Ken. This guy is writing a book to make money. End of comment. All right, last one is from Bob. And this is very interesting. He said, from the Pew Research Center, this is a poll. Americans' trust in scientists, positive view of science, continue to decline among both Democrats and Republicans. Trust in scientists is lower than before the pandemic. You know what I think is really funny? Someone sent me a poll 2 weeks ago that said the exact opposite. It wasn't a Pew poll, of course. It was by someone else. Trust in scientists has risen since the pandemic. So what does that tell you about polls?
That they're poles apart.
So just be careful. I don't know what. Maybe the headline is wrong. Trust in science continue to decline. I mean, anyway, I think the way you do the poll is very telling, and different polls have different results. So, in my opinion, the trust should increase because science has done amazing things in the pandemic. But who is to say that logic and reason should prevail, right?
Not everybody, that's for sure.
All right, let's do some picks.
Yep.
Dixon.
Oh, thank you for allowing me to go first, as usual. And I really apologize for my first pick because it's habit forming. It was for me, for sure. It's 2 hours long, but it's a single Mandelbrot figure that is then explored as much mathematically as you can over a 2-hour period. And the music, Vincent, it's right up your alley. It's electronic music, it's hypnotic, and you get stuck on your monitor and you can't let go of it. It's just remarkable. I haven't seen it all. I skipped, fortunately, to various parts. It keeps gaining in complexity. I wouldn't call that virulence. I would call that fitness. And the Mandelbrot set becomes extremely fit at the end of the 2-hour period. Let's just put it that way.
All right.
My other pick is a climate pick, which I promise to make every pick. from now on. And this one involves the Al Gore Climate Committee on Truth. I don't think I'm saying that correct, but Al Gore is now in charge of a special select committee that investigates greenwashing, basically. And the latest COP28 climate summit was hosted in the UAE in Dubai. And right away, it raises serious flags to, are these people serious about doing something about climate change, or do they just want to make a lot of money, as Vincent would say, by holding a conference in their country and inviting in the world? And apparently the second was more true than the first, because as the UAE was claiming to be all in favor of reducing our carbon footprint, they increased theirs by 20% over the last year, which was quite interesting, actually. And a lot of other countries that Al Gore's committees look into have conveniently, not inconveniently, but conveniently, left out of their tabulation certain greenhouse gases. The complete greenhouse gas was just left out. And they said, well, look, we're lowering our greenhouse gases. Well, of course they are. They're doing it by subtraction. And it's an arithmetic lowering. It's not an actual lowering. They're just playing with the fact that they don't think anybody's going to check them on this. And it's really too bad, because the honesty factor in all of this is really huge. If we're going to do anything, everybody has to be upfront about this. And only the ones that are not being hurt by the concept of eliminating fossil fuels are the ones that are going to be upfront about it. And the others are going to try to protect their interests.
Dixon, didn't you say the introduction, the guy said we should not stop using fossil fuels?
That's right. He says, why should we stop using fossil fuels? They're not hurting the atmosphere. And I have a cartoon that I was going to put in here also. It was from The Guardian, and it's a cartoon of Pinocchio standing in front of a bunch of coal-burning power plants with the smoke coming out. And this Pinocchio's nose is as long as reaching from here to the moon almost, in proclaiming that we've not done any harm to the atmosphere. Just ridiculous. How can people stand in front of other people where everybody knows what the truth is and speak a bald-faced lie and then expect to get away with it?
Because,
and then be insulted
Dixon,
when you start accusing them of lying.
It is money will get you to do anything.
That is true. I hate
Except
to agree
me.
with you.
Doesn't do anything
that's
for
right.
me.
You wouldn't do anything.
Nope.
You asked for it, though.
I wouldn't give you I wouldn't take $1 billion for Microbe TV.
I wouldn't give you my $1 billion, but I might give you a couple of $100 million.
And it's just it's COP28 is just
they should have never held it there. They should have never agreed to that. That was ridiculous. They knew what was going to happen.
Thank you, Dixon.
You're welcome.
Angela, what do you have for us?
My pick is a little bit more lighthearted because it's the end of the year. I just wanted to look at Google searches, and I thought that I would do one that's more like health related. Google search was the top 10, but then just the 100. So this is obviously done by this guy, Brian Dean. He analyzed over 4 billion Google searches, and then they identified like the top 100. Now this isn't science specific. It's just like any Google search. And number one is, what is my IP? I thought was hilarious. How many weeks in a year? So 600, almost 700,000 times. How many ounces in a cup? I've done that so many times. And I know that there's 8 ounces. Like now I know, but past me, or Celsius to Fahrenheit. Oh my gosh, being a Canadian, how to screenshot on a Mac, I appreciate, because I have to teach that to all of my like professors or anyone that I meet that's over the age of 50. How do you screenshot on this thing? How do I do this? When is the Super Bowl is somehow number 5, which I personally don't really care, but for those that like that, okay. When is Easter? When is number 8 was when is Juneteenth? I don't know what that is. What's Juneteenth?
That's the holiday in June. It's June 19th, I believe, and it's the one commemorating the
final slaves finding out that they had been emancipated, I believe, in Texas.
In Texas. That's what I was trying to connect it to.
It became a federal holiday 2 or 3 years ago.
Okay. That I wasn't aware of. But And then if we look at some of the top 10 health trending questions, this is from like CNN. It was on CNN, but they had it. It comes directly from Google. Number one, surprisingly, was how long is strep contagious? It's like strep throat. So I'm guessing there was a lot of strep in 2023. And number two was how contagious is strep throat? Interesting. And then some of the other ones were like, what helps with bloating? How do I lower my blood pressure? The one that I found the most interesting was number 8. What causes preeclampsia? That seems so specific. And like, I'm not sure why, if there was like a spike in preeclampsia in 2023, but preeclampsia is like it's not even just saying, something pregnancy adjacent. It's like very specific. Also, how to stop snoring and how long does food poisoning last? I thought those were funny. But
at the bottom of the list of the Google 100 most asked questions, they sort them out, how many were what questions, where, when, why, or how questions. But the next thing I find really a crack up, because they have top Google questions by type. There were 55 times that people asked what questions, 38 times that people asked how questions. And then just under that, they have percentages. So what questions, 55%. How questions, 38%. If there's 100 top questions, then of course 55 times becomes 55%. It's like, how many times did people ask, how do you convert number of times to percentage?
That's brilliant. It's absolutely brilliant.
That's so funny.
A self-fulfilling prophecy, if I ever saw it.
But yeah. Interesting to see what people ask. That's like when you Google search. Sorry?
Nobody wanted to know what a virus is, huh?
They did ask, what's RSV?
Oh, that's true. That was number 8. That was top 10, wasn't it?
It was pretty high up there.
Yeah.
It was
20,
pretty high up.
top 20.
Exactly.
Yep.
And then right underneath, what is monkeypox? So
yeah,
there you go.
Okay. Those are topical. That's fine. All right. That's great. Very lighthearted. I like that. Thank you. Brianne, what do you have for us?
I have a story that I learned about in reading like a, top 10 science stories of, I read this top 10 space stories somewhere, and I learned so many things. I learned a lot of things that have been going on that I was just not aware of. So there was an astronaut, Frank Rubio, who was at the International Space Station. He actually completed the longest space flight for an American astronaut because he was supposed to be gone for 6 months. There was an issue with the transport back, and so he had to stay there. And so I hadn't really heard a lot about that. But one of the things he was doing during his time on the ISS was he was actually working on growing plants.
Yes.
And so he was specifically growing a number of plants, including some tomatoes, with the idea of researching how one could grow plants in space. And you can imagine all of the reasons why that would be particularly important as we think about longer term space flight. And also, how does gravity impact the growth of all of these plants and things like that. So he's growing tomatoes in space. He picked a tomato to show it to some kids on in a classroom talk, and he went to Velcro it somewhere and do something else, and then apparently he hadn't Velcroed it correctly.
Oh, right.
And the tomato was lost.
That's right.
Rotting, floating around.
And so it was in a Ziploc bag. And so there was this tomato in a Ziploc bag that was lost. And it was like the first tomato they had grown. And everyone assumed he ate it because he had been in space for such a long time and hadn't had any fresh produce. And so this tomato had been lost. Everyone was sure he ate it. He came back. Everyone was asking him about it. He's like, no, I didn't eat the tomato. I kind of wish I did, because then that would be an answer. And so a couple weeks ago, they found the tomato in the Ziploc bag floating around the space station. After
was it like a cherry tomato, or was it like a big tomato? Do we know?
I think it was like a big tomato. They do not tell us about the state of the tomato.
Yeah, I wanted to know.
After it's been floating around, one assumes it was quite dried out because there's not a lot of humidity.
That's right.
But apparently he is innocent of tomato eating. And so I learned both about this lengthy space flight the growing of tomatoes and then this terrible lost tomato that has now been found. And
That's
I
very
was just
funny.
so amused by this whole story.
One of the people who was involved in losing the tool bag was the person who found the tomato, evidently, and exonerated him. So I just have to point out that I had picked a pic that told you how to see the tool bag with binoculars right away after it was lost. And I managed to see the space station, and then with binoculars, I saw the tool bag.
Oh,
so
nice.
it was pretty exciting.
Outstanding.
Yeah.
Oh, that's
Cool.
great.
Kathy, what do you have for us?
I picked something that I've had in my queue for a long time. It's called Earthquakes of the 20th Century, and it's a shorter video that you can watch than Dixon's, and it shows. First of all, you click on the globe spinning icon, and it starts showing you earthquakes that have happened, and the size of the dot corresponds to the size of the earthquake, and the color corresponds to how deep it was, and the globe spins, and then at the end of the time when the globe spins, then they do a summary of earthquakes greater than 6.5, and then they do one of magnitude 8.0 or larger. And so it's just the 20th century. It doesn't include the ones in, for instance, Turkey and Syria recently or things like that. But it's pretty amazing to see where
It's a great
on Earth
video.
they
It's a
find.
great
Yeah.
video.
Yeah.
Cool.
So
Cool.
All right, so my pick is an animator named Chris Georginas. And I want to first explain why I picked this. So yesterday I had a visitor here at the incubator who is also an animator. Her name is Sylvia. She is a big fan and a supporter, and she's an animator for The Simpsons. So she brought me a bunch of gifts, and including this cool poster.
wow.
Which
Cool.
so here on this part it says TWiV forever. She wrote there, and then on the left she drew the girl. The girl. I don't know her name.
I think it's Maggie.
Maggie.
Lisa? Oh, Maggie.
Lisa, right? I don't know.
Lisa's the
Vincent-
older one. Maggie's the baby.
So she
Right.
drew it herself because she's Vincent is my best teacher. Sylvia, 2023. So she brought me this poster, and she also brought me this patch, which was a commemorative patch issued by NASA, and it's got Homer Simpson riding a bull sperm because they did an experiment in the International Space Station on how long bull sperm would last up there. So she gave me this, and I
hilarious.
have
Let's
a
say
hat.
didn't lose that.
I have a hat here
Yeah.
with it.
Wow.
Hopefully it's in a
So
Ziploc
much
bag.
swag.
Ziploc bag. And then she gave me a t-shirt too, right?
Oh, from Springfield. Nice.
Yes, Springfield. Greetings from Springfield, USA, with the fish with the 3 eyes. She explained the story because there's a nuclear plant there or something, and it's going to make mutate. So thank you, Sylvia. But I said to Sylvia, can you do some animations for Microbe TV? I have some great ideas. And she said she will think about it, but she said maybe we get this guy in Boston, and that's Chris Georginas, who's a very nice animator, and he has a YouTube channel that kind of illustrates how to do animations. It's pretty cool. But I want you to look at the tricycle animation study. So he animates a tricycle, and he has his little daughter narrating it. It's just adorable. Okay. You just have to play it and watch. It's really cute. The animation is very simple. It's a white background with just the tricycle with the girl on it going around in circles. And I told her, we just want simple animation, like a virus moving and hitting a cell. That's it. Stuff like that. So that's why I picked George, because he's a cool animator, and he wants to teach people how to animate. So he has this channel, and his name is Keyframer. So Sylvia is a keyframer, right? What is that? You have a character with the hand up here. That's a keyframe. And then the hand is down here. That's another keyframe. She draws both of those, and then she sends the two to Korea, and they fill in all the stuff in between,
Wow.
It's 24 frames per second, right? So for 1 second, they have 24, the two keyframes, and then 22 others. And they outsource the others because it's cheaper to have other people do it, because she establishes the look and everything, and then they just fill it in. So she's a keyframer. And then what is the name she called the other one? I forgot. I'm going to look it up, though. In between keyframes. framer.
But you must be able to do that with AI now, no? That could be generated.
Well, she said there's two ways you can do it. You can in-betweening. So an in-betweener is... So she said a lot of animation is still... She has a big tablet and she just draws. Right? That's The Simpsons are drawn like that. She said you can do 3D animation where the computer draws it and then the computer fills it in and stuff. But a lot of... nobody draws on cells anymore, right? But they still draw, and then they put all of the... They're all digital files, of course, and I shouldn't be explaining this because she should be explaining it. But then the computer just assembles them into a movie, right? But lots of cool stuff. Yeah.
I have an original Simpsons cell in my guest room. Yeah.
So it must be old because they don't do cells anymore.
Yeah, I didn't realize that. Yeah.
She was saying that you would layer the cells on top, and then you do this to see where you draw the next one, right? Because you move it back and forth. And she said that's a very common move to see a keyframer doing this thing back and forth. Anyway, Sylvia was just delightful. She was here for 2 hours yesterday, and she explained this to me, and I showed her all the way we make sausage here at Microbe TV.
I think, isn't The Simpsons around season 32? It's plus 30 seasons now, I think. I think I saw a recent episode and I hadn't seen one in 10 years and I was so confused because so much had changed. But does it, let's see, I need to go.
30 years. This poster is 30 years.
Okay.
And this is 2017, so it's even longer.
Oh, wow.
Yeah.
Okay.
So I wrote her today. I said, you have to autograph it. She said, yeah, I autographed it underneath the girl right there. So I'm going to frame this because I never watched it, but I think it's cool to have an animator. I really appreciate expertise, right? We all do. And animation is an expertise. Sylvia is originally from Bologna. She's Italian, living in Burbank. It's very funny. Cool. Nice to meet you, Sylvia.
Vincent, if you can get The Simpsons to deliver a message that you want people to know, like about vaccination or whatever, that's a very important social media to interface with. I think you should follow up.
Well, she said that Lisa is the most science literate of the...
Well,
yeah.
That's why she put her here.
She should draw another virologist in there like you.
Well, you know what? She's an animator. To get that high-level decision, that's way up there. And then, if it doesn't make money... The Simpsons is owned by Disney now, so it's a big deal, right?
But of course.
It used to be owned by Fox.
Well,
and now it's
a lot of these do have good social messages associated with them, though. I mean, the three-eyed fish next to a nuclear power plant. Come on, that's
right. All right, so we have 2 listener picks. Jason, these have been sitting in my drafts folder for a few weeks. Haven't heard you mention them, so I have a few more links are included. First, a spot-on quick booster explainer that is as much fun to watch as it is informative. So this is something from Twitter, which is a video from Drew, and it's about boosters. So that's cool. And the other is a fascinating article about an inverse vaccine, which shows promise against MS and other autoimmune disease. /dot summary is good. The full article is even better. And Kathy has put in the primary research article, synthetically glycosylated antigens for the antigen-specific suppression of established immune responses.
You should do that on immune.
Should. Yeah. So this would be like to suppress the EBV, suppress the EBV autoimmunity issue, right?
Yes.
Cool. Let's do that. We have a guest next month.
But let's do that.
We'll do it. Thank you, Jason. Is in New York City where it's 2 degrees Celsius. Not anymore.
No.
Not today.
Nope.
And Mako writes,
we're having a heat wave.
Mako writes, hi, super scientists. I think y'all will geek out over this very cool earthquake site for Iceland. It's very cool. It's a map of the earthquakes in Iceland.
Well, that is cool.
Yeah, and cool. I had already put in my link when I scrolled down to see the listener picks and found Micah's. Yeah, it had to be this earthquake one. It's pretty cool.
Pretty exciting a few days ago when there were many 3+ earthquakes in a matter of minutes through the day, but things are a little quieter now. You can toggle for time and magnitude to get a better picture of the current activity. Also, I love Buy Me a Coffee button on the right. A very nice way to say, give me money for my effort. It makes it feel so friendly to give. Maybe TWiV et al. Should do something like that. mica. Cheers, mica, like the mineral.
I love that. That's so cute.
So if you go to microbe.tv, we now have a big button at the top, donate to Microbe TV today, which I've wanted for years to get there. I don't know. Maybe we should say
Oh, there you go.
Give us a coffee. Buy us a coffee, right?
Yep.
Be friendly.
We'll talk your ear off.
We need to make it sciencey, like buy us a falcon tube, something like that.
I just want to add something to
Buy
this
us a flask.
earthquake, to the earthquake map of Iceland. Iceland is on the mid-Atlantic ridge, meaning it's one plate is going this way, and the other plate's going this way. And there's a place in Iceland where there's a gap that filled in with water, and you can scuba dive and touch one tectonic plate and then scuba dive over to the other one and touch the other one. And you
it's
can
the most
pretend
like
that you're pushing it
crystalline.
apart with your hands.
That's
It's very
super
clear, the
cool.
water.
Soon there will be 2 Icelands.
Really?
Like
Well,
actually?
because the mid-Atlantic ridge is going to split it right in half.
That was soon. What is soon, Dixon?
Well, a couple of billion years.
A couple
from now, maybe.
100,000
Soon.
years.
Couple
Dixon,
of
you
thousand,
can't,
that's
you
right.
can't use soon to describe that, Dixon.
Well, it's...
Before you were complaining about 1,000 years, saying
no.
that was nothing,
You
and
asked me
now
how
you're
long. How the
saying
hell do I know?
millions.
I'm just a poor little retired parasitologist.
And Angela, when you wanted something sciencey for the button, when I was in graduate school, there was a new cafeteria cafe that opened in one of the science buildings, and there was a contest for the name, and ended up getting named aliquots. So it could be, we could ask them to buy us an aliquot of coffee.
That's very cute. I like that.
All right. That is TWiV 1075, the ultimate TWiV for 1023. That's right. The ultimate would be the last. Isn't that correct?
Correct.
Yes.
Is there another word?
Penultimate.
Well, that's the one before.
That's the second
That's
last.
right. That's right.
Right, the ultimate
The
TWiV.
final?
The
The
ultimate.
final TWiV?
Yeah.
Well, that's not final. We want to stop? Should
Well,
we
no.
quit?
No.
It's the last TWiV of the year.
Last TWiV of the year, the ultimate TWiV of the year, 1075. You can find the show notes at microbe.tv/TWiV. You can send questions and comments to twiv@microbe.tv. And please buy us a cup of coffee. Go to microbe.tv/contribute. Dixon de Pommiers at Racaniello.org, thelivingriver.org. Thank you, Dixon.
Happy New Year, everybody.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun, and it was a fun year of TWiV.
Fun
absolutely.
year. It was. We did a lot of TWiVs, at least 52 in the 52 clinical updates, and then I think there weren't any extras, maybe just a few.
Yeah, I thought it looked like it went from 971 to 1075 in my file folder.
That's right. We hit 1,000 in April. That was fun. Brianne Barker's at Drew University, BioProf Barker on social media. Thanks, Brianne.
Thanks. I learned a lot, and Happy New Year, everybody.
Angela Mingarelli is at McGill University, Immune Vet on social media. Thank you, Angela.
Thank you, guys. And I am excited for a new year, 2024. Happy New Year, everyone. See you in the new year.
And in fact, Angela, next week we are going to release the McGill episode.
Okay, good. I'll let everyone know.
Because I'm away I'm at Princeton for Thursday and Friday for working on the book. So we won't have a recording. So I will release our cool McGill with 1, 2, 3, 4, 5, 6 participants, right?
Yeah.
It was fun. It was a lot of fun. I'm Vincent Racaniello. You can find me at microbe.tv. I would like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.